Look at this single drop of blood. As it shifts from a healthy crimson to this murky, lipid-heavy state, we are seeing the transition from simple energy storage to systemic toxicity. Clinical models now define obesity as an active, chronic, low-grade inflammatory and endocrine disease. In patients with a body mass index between 30 and 34.9, this inflammatory state is linked to a four-fold increase in the risk of concurrent myocardial infarction, stroke, and type 2 diabetes. To understand these comorbidities, we have to track the pathology from the expansion of the individual fat cell to its distant systemic endpoints. This inflammatory cascade targets three networks: the endocrine pancreas, the cardiovascular system, and the brain. Effectively treating cardiometabolic multimorbidity requires a move away from managing isolated symptoms to address the root of systemic inflammation. The pathology begins within the adipose tissue. During chronic overnutrition, fat cells expand to accommodate excess triglycerides. In this cross-section, the localized structural limits are visible. As adipocytes enlarge, they outpace their vascular supply. The capillary network stretches and fails, leaving the center of the tissue mass deprived of oxygen. This oxygen deprivation triggers the expression of hypoxia-inducible factor 1 alpha within the tissue. HIF-1 alpha upregulates chemokines like MCP-1, which act as a chemical distress signal to the immune system. The local immune environment shifts. Resident anti-inflammatory M2 macrophages are replaced by pro-inflammatory M1 macrophages that form crown-like structures around dying cells. These structures produce a constant output of tumor necrosis factor alpha, interleukin-6, free fatty acids, and the immune protein complement C3, all of which enter systemic circulation. This localized oxygen crisis converts adipose tissue into an endocrine organ that pumps inflammatory mediators throughout the body. These mediators travel through the bloodstream to the pancreatic islets, the clusters responsible for blood sugar regulation. At the molecular level, the impact on the pancreatic beta cell is direct. Circulating free fatty acids bind to toll-like receptors 2 and 4 on the cell membrane, sending a chemical signal to the nucleus. This signal activates the NF-kappa B pathway, which impairs the cell's ability to express the insulin gene. At the same time, adipose-derived complement C3 arrives at the islet, activating WNT beta-catenin signaling within the cell. Under this dual stress, the beta cells undergo dedifferentiation. They shed their insulin-producing identity and revert to a progenitor-like alpha cell. The progression of type 2 diabetes in these patients is not merely exhaustion, but a forced cellular identity shift driven by distant adipose signaling. The cardiovascular system faces the same inflammatory stress, beginning with the inner lining of the blood vessels. This cross-section shows the resulting vascular damage. Systemic cytokines force endothelial cells to express adhesion molecules, allowing circulating monocytes to pull themselves into the subendothelial space. Once inside the vessel wall, these monocytes use scavenger receptors to engulf particles of oxidized LDL cholesterol. This intake transforms the macrophages into foam cells, initiating the formation of atherosclerotic plaque. Beyond the vessels, the heart itself undergoes structural remodeling. Increased adipose volume load forces the heart to thicken, leading to a stiffening of the ventricle known as heart failure with preserved ejection fraction. This schematic reveals how a localized fat deposit accelerates this failure. Epicardial adipose tissue presses against the heart muscle and secretes cytokines directly into the tissue. This crosstalk between fat and muscle drives the development of interstitial fibrosis, making the heart muscle rigid and scarred. This scarring reconfigures how heart cells express ion channels. These conduction blocks disrupt the heart's rhythm, resulting in atrial fibrillation. Obesity dismantles the heart's mechanical capacity and its electrical architecture, making heart failure and arrhythmias common clinical endpoints. Systemic inflammation also permeates the blood-brain barrier. Neuroimaging from the Life Adult study has tracked the exact impact of this inflammation on the brain. The imaging shows a direct correlation. High visceral fat volume is linked to accelerated microstructural brain aging. This includes a loss of connectivity in the default mode network, gray matter atrophy, and the development of white matter lesions. Within the hypothalamus, the adipose-derived hormone resistin initiates a specific disruption. Resistin triggers central insulin resistance, which compromises the brain's ability to sense glucose and satiety. This resistance creates a self-reinforcing cycle. The structurally altered brain chemically promotes the consumption of more calories, worsening the underlying disease. Clinical data sometimes shows a phenomenon called the obesity paradox, where moderately obese heart failure patients outlive their lean counterparts. This survival advantage appears to contradict the pathological model of adipose inflammation. The resolution lies in the limitation of body mass index. BMI is a monolithic metric that fails to account for how fat is distributed. This model highlights the functional difference between fat types. Subcutaneous fat, found under the skin, provides a metabolic reserve during the stress of chronic illness. It secretes adiponectin, which has anti-inflammatory and cardioprotective properties. Visceral fat, located deep in the abdomen, is the primary driver of the M1 macrophage and complement C3 cascade. The obesity paradox is an artifact of measurement. BMI masks the distinction between where fat is stored and how it biologically functions. The changes in the pancreas, the heart, and the brain all originate from a single source: hypoxic visceral adipose tissue. Cardiometabolic multimorbidity is a unified pathology. Therefore, effective management must move beyond mitigating downstream symptoms to resolve the root systemic inflammation and metabolic inflexibility.